Hi, speaking on the line is Novelty Fatime and Giang, and today I will show you and discuss what is DNA biotechnology. If I will just ask you about what is DNA biotechnology, you will probably say about the blueprints for life or the genes that we acquire from our parents. So now we're going to identify what is really DNA biotechnology. So before that, buckle up your seatbelts and get ready for the lesson. What is DNA biotechnology? So when you hear about biotechnology, maybe you think all about the news such as DALI, the cloned chip, genetically modified organisms, or the therapy used in medicine such as the stem cell therapy for human knee joint. DNA biotechnology is a modern biotechnology that uses a scientific research on DNA for practical benefits. DNA biotechnology is synonymous with genetic engineering because the genes of an organism are changed during the process and the DNA of the organism is recombined. While genetic engineering is the deliberate modification of the characteristics of the organism by manipulating its genetic material. How does genetic engineering work? So as we all know, genetic engineering is a cornerstone of modern biotechnology, and it is based on the scientific tools developed in recent decades that enable researchers to identify the gene that produces the protein of interest, cut the DNA sequence that contains the gene of a sample from a DNA, placed the gene into a vector such as a plasmid or bacteriophage, use the vector to carry the gene into the DNA of the host cells such as E. coli or the mammalian cells grown in culture. Induce the cells to activate the gene and produce the desired protein. And lastly, extract and purify the protein for therapeutic use. So by the existence of genetic engineering, insulins for diabetes have produced using the genetic material of a certain organism. And later on, I will discuss and show to you what is the process of genetic engineering through DNA cloning. Here are the applications in DNA biotechnology. DNA profiling, DNA cloning, Transgenesis, genome analysis, stem cells engineering, xenotransplantation. So our first stop is the DNA profiling. What is DNA profiling? So DNA profiling is the process where a specific DNA pattern called a profile is obtained from a person or a sample of bodily tissue. Here's a picture of a polymorphism or the singular nucleotide polymorphic, which has adenine, guanine, and thymine. DNA polymorphisms can be analyzed to give a DNA profile. So even though that we are all unique, most of our DNA is actually identical to other people's DNA. However, specific regions vary highly between people. So these regions are called polymorphic. So the differences in the variable regions between people are known as polymorphisms. So each of us inherits a unique combination of polymorphisms from our parents. Again, DNA polymorphism is a DNA sequence variation that is not associated with any observable phenotypic variation and can exist anywhere in the genome, not necessarily in a gene. So polymorphism means that one of two or more alternative forms, alleles, of the chromosomal region that either has a different nucleotide sequence or it has a variable numbers of tandem repeated nucleotides. So DNA profiling has a can be utilized also in identifying the probable origin of the body fluid sample associated with the crime or the crime scene by the use of DNA fingerprinting, for example. Then next is the revealing of family relationships through DNA testing. So as you undergone the DNA testing, genetic codes shown in the picture will reveal the family relationships. For example, you are the missing daughter of that person, or you're the missing son of the most notable personality. The next one is the identify disaster victims, especially the remains. Then, now that you already know what is DNA profiling, let's have a checkup test. So, who do you think is the killer in the crime scene? So, here's the crime scene. The suspect 1, 2, and 3. So, if your answer is suspect number 2, then you are correct. Since the codes shown in crime scene has the same codes in the suspect number 2. So it's just easy to know who is the killer in the crime scene. So congratulations, because you already know what is DNA profiling, especially in the crime scene. Next is the DNA cloning. DNA cloning is the process of making multiple copies of the particular segment of DNA. It is also called as recombinant DNA technology. So as what I've promised a while ago, I will show to you how does a DNA cloning work. So here's the picture. This is the process of DNA cloning. So the first step is that the selected DNA fragment is inserted into the plasmid. So this is the plasmid, the circular piece of DNA using enzymes. Then the next one is the restriction enzymes and DNA ligase are used in the process. So it was like a pair of scissors and a glue. So the restriction enzymes are used to cut, which serve as a pair of scissors. The DNA fragments at specific sequences, and then the DNA ligase are used to join the nicks, which serve as the glue. So here is how it is cut and glued. Then the third one is the recombinant DNA thus produced is introduced into bacteria. This bacteria produce an exact copy of the plasmid. So here's the bacteria inserted, and then it makes a copy which known as the clones. So what is cloned DNA used for? DNA cloning is used to create a large number of copies of a gene or other piece of DNA. The cloned DNA can be used also to work out the function of the gene, investigate the gene's characteristics such as the size, expression, tissue, and distribution. Look at how mutations may affect a gene's function. Make large concentrations of the protein coded for by the gene. Actually, there are two types of DNA cloning. The first one is reproductive cloning. It is a process of making a genetically identical copy of an organism. Here's an example. We have here a somatic cell with the SAR gene, this one, and another cell which is the egg cell. So the nucleus in the somatic cell has been removed, same as the nucleus in the egg cell. However, in the egg cell is being used without the nucleus, which have been removed. However, the nucleus from somatic cell has been fused with the nucleated egg cell. Then later on, it produces a clone. The second is the therapeutic cloning. So it is the process of making multiple copies of a cell to treat a disease. Here's the picture again. So the nucleus is taken out from human egg cell, is replaced with the nucleus from one of the patient's cell. The next, the egg cell is triggered into an embryo. Then after 3 days, the formation of cells create some subdivisions of it. Then after 5 days, cells are removed from the embryos with a pipette, this one. Then after that, stem cells from embryo are grown in the dish containing nutrients. Then, lastly, stem cells develop into different tissues and organs. This can be used for medical treatment. So, for the next topic, we will also discuss about stem cells. So now that we are already done about DNA cloning, let's now have again a checkup test. So let's arrange a sequence of how DNA works. DNA cloning works, rather. So what do you think comes first? So if your answer is DNA fragment inserted into a plasmid, then you're correct. The second one is the restriction enzymes are used to cut the DNA fragments at specific sequences. The third one is the DNA ligase enzymes are used to join the nicks. The fourth one, the recombination of DNA thus is introduced into the bacteria. Then the last one is the bacteria reproduce and produce an exact copy of the plasmid known as the clone. So good, because you already know how the DNA cloning works. Let's go on to transgenesis. What is transgenesis, by the way? Transgenic organisms contain foreign DNA that has been introduced using biotechnology. We have examples such as the transgenic mice are developed by injecting DNA into the oocytes, or 1 to 2 celled embryos taken from the female mice. We have also the spider goats that are obtained by the pronuclear microinjection and somatic cell nuclear transfer. Here's an example of how transgenesis works. We have a desired gene, the gene for milk protein, and a foreign gene. Then, they were combined. And then, the injection of a foreign gene into an embryo, the single-celled stage, this one. Then, after that, the embryo will be the transfer of the embryo to the uterus of the female animals, such as the goat, the rabbit, the mouse, the pig, the sheep, and lastly, the cow. So the aim of transgenesis is resulting transgenic organism that will express the gene and exhibit some new property of characteristics. So we have transgenic techniques also. Number one is the DNA microinjection. So DNA microinjection is a method that involves the direct microinjection of a chosen gene construct. So a single gene or combination of genes from another member of the same species or from the different species into the pronucleus of a fertilized ovum. So as you can see, it uses a microinjection directed to the nucleus. Number two is the retrovirus mediated gene transfer. Retroviruses are commonly used as vectors to transfer genetic material into the cell. So as you can see in the picture, you can see this is a cell. Then a microinjection also applied, but this time vectors is used to transfer genetic materials, which this purple color, this is the vectors. Yes. Then the offspring derived from this method are chimeric. Not all cells carry the retrovirus. So the transmission of the transgene is possible only if the retrovirus integrates into some of the germ cells. The third one is the embryonic stem cell mediated gene transfer. In this method involves prior insertion of the desired DNA sequence by homologous recombination into an vitro culture of embryonic stem cells. So we have here a clone DNA, clone DNA, and then injection of the transfected embryonal stem cells into the normal mouse. This is the normal mouse. So once na it was injected, there is an implantation. Then once the chimeric mouse is mating with a normal mouse, they will produce an offspring which is a hybrid. Now let's go on to genome analysis. Genomic analysis is the identification, measurement of comparison of genomic features such as DNA sequence, structural variation, gene expression, or regulatory and functional element annotation at a genomic scale. The tools of DNA analysis include enzymes and fundamental procedures which allow amplification and detection of specific targets on the genome and the construction of the genome libraries using recombination techniques. Gene expression analysis is most simply described as the study of the way genes are transcribed to synthesize functional gene products, functional RNA species, or the protein products. So the study of gene regulation provides insights into a normal cellular process such as differentiation and abnormal or pathological process. We have also here the picture of how gene expression analysis works from a sample to the sample preparation and the sample assessment in which the testing will be conducted. The next is the reverse transcription. The sample will then transcribe and then profiling and quantification until it goes on to the gene expression profile. So here we now on the stem cell. So what is stem cells? So stem cells are undifferentiated cells that are capable of self-renewal or differentiation into a specialized cell. So they exist within areas of the body known as the biological niches and are essential for growth during childhood and homeostasis throughout adulthood. So as you can see in the picture, all of this is present in our bodies. So there are two sources of stem cells. The first one is the embryonic stem cells. The embryonic stem cells are found in the inner cell mass of the blastocyte post 5 days of development. So these cells are potentially immortal and are derived from embryos before they implant themselves in the uterus. These cells are typically 4 to 5 days old and are called blastocysts, a hollow microscopic ball of cells. So imagine, when you were still a zygote, you were first part of the embryonic stem cells. So the next one is the adult stem cells. Adult stem cells are undifferentiated cells that are totipotent and pluripotent. When you say totipotent and pluripotent, they are all potential in our body that can develop into all types of cells in the body. So for example, the zygote formed during fertilization of an egg. So the first few cells after division. It also come up with the division or the renewal of the cells. The key applications of stem cells. So first one is the therapeutic purposes. So in therapeutic purposes, apart from bone marrow transplantation, stem cells used in therapeutic also because it attempts as being made use in treating type 1 diabetes, Parkinson's disease, cardiac failure, and so on. The second one is the skin replacement. stem cells are currently being used to grow skin from the black hair of a patient. The hair follicles contain skin stem cells or the keratinocytes, and these cells can be isolated and cultured to grow an epithelial sheet. The third one is brain cell transplantation. Stem cells can be employed to generate dopamine, a chemical that is lacking in patients suffering from Parkinson's disease. However, in some cases, patients develop side effects suggesting presence of very high levels of dopamine. Lastly, we have xenotransplantation. Xenotransplantation is when the living things in a living cell, tissues, or organisms are transplanted between species. So here's a picture of a human heart has been replaced with the pig's heart. So here's the uses of xenotransplantation. We have organ transplants, replacing diseased organs such as hearts, lungs, livers, pancreas, and kidneys. So in the picture shown, it conducts a heart transplantation. The next one is cell transplants, replacing damaged or destroyed cells in diseases such as diabetes, Alzheimer's, and Parkinson's disease. In that picture, it is there's an injection of insulin for the diabetic person. So, tissue transplants, skin grafts, cornea transplants, or bone transplants. We have also here the donor eye and the recipient of the eye, which undergoes the tissue transplants of a cornea. The last one is the bridging transplants, providing organ function externally to patients with organ failure. A picture of a man who has a hard time to breathe because the blood in his heart couldn't pump. So there is a device attached to his heart. And that ends the my topic for today, which talks about DNA biotechnology. Again, I am Novelty Fatima May M. Diang. And now that you have learned about DNA biotechnology, I hope that you realize the significance of DNA as a part of our lives. Because you and I are the products of DNA.